In the 1980s, cells were taken from a male myeloid leukemia patient and used to make the KBM7 cell line. Later, this new cell line was used to generate HAP1 cells. Most normal human cell lines are diploid, which means that they have 2 copies of each gene. If you want to knock out a gene, having 2 or more copies makes this difficult. Since the HAP1 cells are near haploid, they are a good model to use as they only have 1 copy of most genes. Over time, HAP1 cells may revert to a diploid state. If this does occur, your edited gene will still be present and the knockout will not be affected. HAP1 cells also show sustained growth at single-cell dilutions, are relatively easy to transfect, and have a rapid doubling time. There are many additional advantages to performing a knockout using HAP1 cells. For example, HAP1 knockouts are a relatively affordable knockout model, and also a widely applicable model. We have HAP1 knockouts for over 7,500 genes currently, and new knockouts can be made on request. A common misconception about knockouts is that the entire gene is removed, like this. But this isn't right. Instead, HAP1 knockout cell lines are made using CRISPR. A guide RNA is designed, usually to an early exon. This guide directs Cas9 to cut, making a double-stranded DNA break. The DNA break is repaired by the cell's endogenous repair machinery in a process called non-homologous end joining, or NHEJ. NHEJ is an error-prone repair mechanism that often results in small insertions and deletions called indels. We isolate clones with frameshift mutations, which usually lead to a premature stop codon. We confirm the presence of the frameshift mutation at the DNA level and provide you with the sequencing information and the primer sequences. Let's look at exon 3 in more detail. Here's the DNA of exon 3. The guide binds here through base pairing with the nucleotides in green. Cas9 cuts the DNA at the bases immediately adjacent to the PAM site, which is marked in blue. The DNA repair mechanism of the cells will then be activated and leads to the creation of small indels. Indels can be small deletions like this, or they can be small insertions like this. When making HAP1 knockout cell lines, we isolate clones with frameshift mutations, like this one. Here, insertion of one base leads to a frameshift. The frameshift usually causes a premature stop codon. Here's the wild-type DNA sequence, which gives the wild-type amino acid sequence after translation. Here's the sequence with a one base pair insertion, which causes a frameshift. When we make HAP1 knockout cells, we validate the presence of the frameshift mutation by PCR amplification and Sanger sequencing. In this example, translation of the sequence leads to a short novel sequence, shown in orange, and then a premature stop codon, shown in red. This can lead to the absence of a protein or to a non-functional protein. If you have any questions, please feel free to reach out to us at technical support by email, phone, or chat.